So, good morning, everybody, and welcome to one more day of our event here. And today we will start with an important topic. Is about antimicrobial resistance, and we have 3 lectures for today. And I would like to present the first one for everybody. The name is Casey Jung, Dr. Casey Jung. He's from University of Florida, and I did my training with him. And I appreciate your presence here, Dr. Jung. So I want to invite him to present for you about a little bit about AMR. And that's it. I would like to call you to the stage. Thank you so much to come here and enjoy.
Good morning, everyone. It's a little bit tired because I had about 3 glasses of wine. And I decided to change one of the chapter, one of the lesson last night. So, because I wanted to deliver some unpublished data, because if I put already published data, you can read all of the information, from the PubMed, from my webpage. So I wanted to deliver some new data. It has not published yet. So if you find some typos, errors on my slide, please excuse me. So I made it after 3 glasses of wine last night. So I slept 1 a.m. and I woke up at 5 a.m. again to finish this talk. Okay, please. So Marcos. So I like to review this pilot. What this mean? Oh my gosh. Yes, do and this one. Yes, do something to win. Yes, yesterday the first pillar was study, to know. Yes, so I think this is the beauty of doing seminars, presentation in a row. So I can use my own data to educate or to review whether you were attending this one or not yesterday. Marcos was not here yesterday, So we visited to study, to know. Now we're going to do something to win against antimicrobial resistance. Okay. So today's title of today's talk is AMR in humans and animals. And I wanted to talk about many dissemination mechanisms of AMR in humans, animals, even at the interface of humans and animals. So again, this is my course outline. First, I wanted to introduce AMR and AMR in human hospitals and AMR at the human and animal interface. And I want to conclude with some questions. So do not sleep, okay? Wake up. So AMR. So, yes, I'm sorry to starting with this very different slide. Back in 2013, we had about 2 million people infected by AMR, and 23,000 people, they died. But in 2019, after 6 years later, the numbers, what happened? Numbers increased to 3 million, almost by 50%. Also, death toll is about 36,000 people, which is equivalent to the number of total people died with car accident in the USA. It was about 43,000 people killed in 2011. So how many times have you seen people died with car accident? I saw multiple times killed by car accident. So this is not rare. This is very common happening in human hospitals. And the case was even worse during pandemic. The infection raised about at least 15%. In 2019, 2020, but the most problematic is these guys, carbapenem-resistant Acinetobacter, Candida, even ESBS, increased about almost 80%. Because those pathogens, they are very notorious, and almost we have no options to cure them if I am infected by these bacteria. So this is a 2014 estimate. And they estimated in 2050 we may have about 10 million people globally killed by AMR. I believe you saw these pictures a lot because many people talking about AMR, they cited these studies. And these are the bacteria they pose the greatest risk. So my lab is working on these guys with Shanghai Hospital with MDs, doctors, and then I work with these guys for some food safety related or animal infection or related diseases. So this one is mainly prevail in human hospitals, and these guys are in both humans and animals. Not only human hospitals, but also animals, they get sick and they get treated with antibiotics. Therefore, we also have antimicrobial resistant bacteria on animal farms. So not only that one, before 2017 in the USA, we used some antibiotics without prescription for prophylactic purpose to prevent animal disease. And sometime, since the early 1950s, we used some antibiotics for growth promotion purpose, even though the effect of the growth promotion purpose was debatable. So many people used because we believed it maybe increased the animal growth rate a little bit. And unfortunately, many people use this information about 70% of the antibiotics produced in the US being used in agricultural settings, not only animals, also plants. However, if you divide the amount of type of antibiotics being used in the US especially, it's a little bit different. Excuse me. So in the US, main antibiotics being used is tetracycline in animal farms, also apple orchard, agricultural settings, and also macrolides. Those are the main antibiotics being used in agricultural settings, but not frequently used in human hospitals. The most important antibiotics being used in medical setting is cephalosporins, especially third and fourth generation cephalosporins. They are not used in here. Only one cephalosporins prescribed for animal treatment purpose is ceftiofur. That's the third generation cephalosporins. That's beta-lactam antibiotics. But other than that, there's no FDA approval at this moment. But many people, they blame animal producers, plant producers. You guys are the kind of people who raise AMR all the time because of these numbers, 70%. Okay? So maybe we have to, think about this. Whenever some people say about these numbers, we have to proactively talk to them. Maybe 70% of total, but it's not used for human medicine. Okay, not only this one, also this, not only the number of antibiotics being used in agricultural settings, but this one, colistin-resistant antibiotics in 2015 and 2016. It was very, very hot news because people said the end of the road is very far away for antibiotics because of this. Colistin was developed in early 1950s. It was very effective antibiotics, but we didn't use in human hospitals because of huge side effects. If we take it, it may damage our kidney. Therefore, we didn't use it in human hospitals. But unfortunately, including your country, Brazil, Canada, China, we heavily used these antibiotics for animal production purpose. And it alarmed us. Transmission of AMR produced in agricultural settings come to human hospitals. And we lost one of the most important antibiotics for human treatment purpose. Okay? So now everybody believes, okay, this is also reviewed by my lab. We have studied ESBL's extended spectrum beta-lactamase for a long time, and then we wrote review papers. It found that the ESBL genes happens everywhere, wildlife, human, manure, soil, water, air, and they transmit without stopping. This is a vicious cycle. Not only us, So now everybody believes, One Health, maybe that is the key to stop or slow down AMR transmission between different niche environments, animals, and people. Then we may have better human health. So to summarize my first lesson, I would like to use this cartoon. This is the cycle of resistant bacteria. It happens everywhere. Human hospitals, maybe humans, they shed antibiotic resistance through the sewage systems, also water irrigation systems, and then they circulate here. And then sometimes they are leaked into the environment. Fish and cows are there drinking waters, and then people, he only eat tilapia. Maybe he got a lot of AMR from fish. And these cows infected with AMR is shed, and then it goes to the produce, and then it go back to again fish, cattle, swine, and then come back to humans. So this is kind of common sense nowadays. We all agree, right? That means we are exposed to antibiotic resistance all the time. Not only me, animals, fish, everything. So that raised huge concern that, I may get infections sometime soon. Or I could be one of the person, out of 35,000 people in the US, maybe die with these antibiotics. Please keep this in your mind. I may revisit at the end of this talk. Okay? And then in lesson 2, I wanted to talk about AMR transmission in hospitals. So using NDM. NDM is one of the notorious antibiotic resistance. And it only happened it started about 20 years ago. It started in Delhi, India, because of the name. It came from, we first identified this resistance, beta-lactamase in India. And it gives resistance most of the carbapenems. That's one of the most important antibiotics for human medicine. But still, we don't know why this NDM spread so fast. And there's also some lacking information about that, and we hypothesized that maybe they have multiple mechanisms to transmit these NDM genes or plasmid encoding these genes. So we wanted to understand the occurrence and transmission of NDM pathogens in hospitals and to identify mechanisms and NDM genes spread in healthcare settings. So we collected about 11 NDM pathogens at University of Florida hospitals. We call it change hospitals over the year. So we don't have a lot of cases because normally I receive strains from change hospitals when they fail to treat patients infected with bacteria. Once they cure them, they don't worry about it. So they do not send the strains. 11 very dangerous strains. They did all different types of treatment. They failed, and they gave us to understand what's going on with these guys. From 9 different patients. And this is antibiotic susceptibility test we did. All of them are multi-drug resistance. Not only metophenol, but some guys, they are resistant to colistin and many different cephalosporins, also beta-lactams, and other different types of antibiotics. And then we did a whole genome DNA sequencing first using Illumina. And what we found is this. We found Enterobacter hemorrhagic from patient B and patient A. They have quite different genetic relatedness, distances, so they are different strains. And then Klebsiella pneumonia, they have very few number of SNPs, about 14 and 18, so we believe they are same strains, showing they are clonal transmission because it was isolated from patient C and patient D. And another one is E. coli. It was isolated to patient C. They all carry NDM genes. So I see clonal transmission of these strains between patients. Then we decided to spend more money to fill the gaps of the sequencing. So we used the PacBio. It's a little bit expensive, but we did it to understand even further. Especially we look at, look at this strain's Klebsiella pneumonia. It was in patient C and patient D. And we wanted to try to understand where these patients got these strains. And this is the kind of, patient B, A, C, D, they were housed in our hospitals for about 140 days. Especially C and D patient, they were housed in burn unit at our hospitals. And this is the isolation date. They were not infected at the same date, but they were housed in the same unit. Therefore, we assume that these patients, they got the strains from the burn unit. And then we wanted to know, how does plasmid transmission also spread the NDM genes? And we look at this plasmid, PKC45K1 and PKC45K5. They are almost identical except very small reason. The size of the plasmid is 45K. It's a huge, humongous plasmid, but it was isolated in Enterobacter hemorrhagic strains from A and B patient. But the strains are different, but plasmids are the same. That means plasmids were transmitted or transported between two strains isolated to two different patients. And we wanted to know how this plasmid transmission happened. But the font size is too small. But this plasmid carried the Trojans, which is necessary for the conjugation of the plasmid DNA. These plasmids were isolated from different genus, E. coli and Klebsiella, and even though from different patients. And then this strain was also very prevalent in other strains, other genuses we isolated, we downloaded from NCBI public database. So this plasmid somehow has a very good capability of persistence. And this is a conjugation mechanism, but I don't think I have to visit this. I think everybody took the microbiology course to understand the conjugation. And we measured the conjugation of frequency. This plasmid was given to E. coli, exceron blue, this is the recipient. The conjugation frequency was pretty high. About every single strain in isolate out of 10,000, they could have got the plasmid from Klebsiella pneumonia. It shows that this plasmid was transmitted to another strain under the genus through conjugation method. And then I wanted to bring another mechanism, which is IS element biotransposon. So IS element is also very famous mechanism for gene transfer. So this gene was in the chromosomal DNA, but once they received an IS element, they could go to the plasmid, and then they could go to conjugate plasmid. It is intracellular mobility. Then if they are encoded on conjugation plasmid, they can go to another strain. It's intracellular mobility. So this is the mechanism, these NDM genes are transferred, and we wanted to know. And we found that this is the NDM genes. It was coded by IS30 family, also IS19 family, and they have exactly the same sequences. Therefore, we believe this IS element is jumping between different genus. E. coli and Klebsiella and Enterobacter, they have exactly the same insertion sequences and IS families. So this is kind of a summary of NDM gene transfer. It is encoded by IS element first, and then it went to the plasmid. And this plasmid itself, they can transport other genus. Also, this bacteria itself, they can transfer to other patients. Therefore, this bacteria and the gene have 3 different mechanisms of transfer. One is clonal transmission, means bacteria itself transmits to other patient. And we could see conjugation mechanisms. Also, we could see IS element. So this is the kind of perfect storm why these NDM genes spread very quickly in human hospitals. It's very scary, isn't it? So now I wanted to move to the interface between humans and animals. I believe I scared you guys already too much. Now I'd like to give you a little bit positive information. So we want to know whether really bacterial transmission happens between humans and animals. In the previous lesson, I just explained that happens in human hospitals where a lot of sick people there, also a lot of immunocompromised people are housed. And that's the reason why they are susceptible to transmission of this bacteria, especially for colonization. So we wanted to know whether AMR pathogens really colonize in multiple hosts without specificity. And also I wanted to know, they really break host barriers, is the same question. And with comprehensive knowledge of genetic features, maybe help us to understand how AMR pathogens adapt in different hosts, in this case, human versus animals. So we used MRSA as a model organism because MRSA is very famous. Everybody knows what is MRSA because MRSA, people get infected by nosocomial infections in hospitals. Also, people get infections in community. Also, animals are infected by this MRSA. So everybody think, oh, MRSA, it has a huge capability to infect everywhere, everyone, humans, animals. And sometimes some people believe this is a human pathogens because after taking some food, they get infections. But there's no agreement about that. So we call it, hospital-associated MRSA is HA, and community-acquired is CA, and livestock-associated is LA MRSA. And this is very well-accepted cartoon showing MRSA, they can circulate everywhere, hospitals, meat process facility farmers, also meat and communities, also environment, they survive very well. And go back to the livestock and humans and so on. So it looks like there's no point to stop this spread of MRSA. And also MRSA forms biofilm. That means it's so hard to get rid of. Valentina shared these ideas yesterday. Yesterday? Yeah. 2 days ago. So MRSA, not only they have super duper good capability to infect everyone, but also maybe that's the reason why we have MRSA spread everywhere. Oh, Brazil, oh, here. But not that high, I'm sorry, this is the hottest area we have, I'm sorry. And USA, everywhere. South Korea also hot. Canada, some of the countries are not shown because we do not have data in Africa. Okay? So MRSA is everywhere. So this is the best strain to study whether MRSA can infect everyone without any restriction. And also we know that MRSA is a generalist. Generalist means they can infect everyone. So based on this clonal complex study, we can see MRSA clonal complex, these guys, they infect everything. But some clonal complex like CC25 specialized for humans, and I don't know what is this. Also, this one is for cattle and sheep. So we see 2 types. One is generalist, they infect everyone, but some guys, specialists, they have very narrow range of habitat and specific environmental fitness. Maybe because they are recognized as specialists because we do not have enough numbers, enough samples. That's truly possible. Therefore, we wanted to know whether MRSA is a true generalist or not by using machine learning approaches. Because At the time when we started this work about 2 or 3 years ago, at the time sequencing is the best technology we could have. And with that information, we had some limitation to understand this. Therefore, we used this. And this work is, again, as I said in the beginning, is not published yet. Please do not share. And this work is funded by NSF and U of Florida, also National Research Foundation of Korea. So we collected about 2,000 genomes of MRSA around the world. So mainly focused on in the US and China and European countries because not only they have more cases, but also they have more monies to sequence them from different sources. This is the genome content. I would like to skip it. And this is the phylogenetic tree of more than 2,000 genomes of MRSA. Okay, your computer cannot generate these trees. U of Florida, you have HyperGator is a supercomputer. It belongs to top 5 supercomputer in the USA, in academia. So by looking at this, if we isolated MRSA from humans, is this color, and non-humans in this blue. As you see here, there are some blues and some, what is this color? It's whatever, right? But looks like we have some interspersed clusters here, but some of them are here. So it's not clear whether MRSA is a generalist or not. But when I added MLST data here, like ST5, sequence type 5, we see humans and animals, they were interspersed. So it looks like this area is generalist. And if I look at this area, especially ST22, all of them are human isolators. And interestingly, this area, it is belong to ST398. I'm sorry, I forgot the name. So I'm sorry, these guys, again, is the same clusters, the same MLST, but this region is non-humans and others are humans, even though they belong to same ST types. Okay. With this information, the most advanced technologies at the time, whole genome DNA sequencing, SNP analysis using and then phylogenetic trees using supercomputer, it doesn't provide clear ideas. So we decided to use even better one. I want to skip this, which is machine learning. Yes, yesterday I said that we started to use machine learning. Because of the power of machine learning, we started to see something. It was not revealed before. And also it has a high scalability, which is also very fast. Also we have improved accuracy. Since we have many students attending this seminar, I decided to put some basic information about the machine learning to educate you guys. So machine learning is under AI. There are machine learning and data mining, but we don't have capability to do data mining yet. But machine learning, we use mainly supervised machine learning. Sometimes we use unsupervised machine learning, but mainly supervised machine learning. Supervised means when we put input data, we tag them. Oh, these guys came from humans, these guys came from animals. But when we use unsupervised machine learning, input data, we do not provide any information, and then ask machine to identify the intrinsic factors, intrinsic patterns. Then they keep running it to provide this kind of data. So I'm not going to talk about unsupervised machine learning a lot, but today supervised machine learning. Supervised machine learning has 2 groups based on the outcomes. One, many machine learning like SVM or neural network or random forest, this is a classification. And sometimes we have outcomes shown as regression models, like linear, polynomial, or logistic. We use the regression model, but many people use classification. Data will be sometimes binary, like yes or no, 1 or 0, vaccinated or not, something like that. Yesterday I said, this guy's from 3 months cattle, 9 months cattle, 18 months cattle, or microbiota. Machine could predict that accurately. So we use support vector machine to group them whether this genome was originated from humans versus non-humans. A little bit more information about the support vector machine. So we wanted to separate whether they came from humans or non-humans. And the best way is, SVM model, they provide optical hyperplane to separate those two groups. To do this, machine recognized the maximum margin and support vectors. So this maximum margin is kind of these guys are very nascent to adjust to this maximum margin. Therefore, these guys are used to support vectors, and these humans are support as a vector for the margin for the human isolators. And what we did is very simple. Honestly, it's not very simple. So 2,000 genomes, humans, non-humans, we identify all of the genome presence, gene presence and absence using RORI. Also, we measure the significance using SCORI. They are under Python. So if you use Python, you are familiar with this information. And then we tagged them, because it's a supervised machine. It's from human, it's not from humans. And then we generate the data matrix, and then ask machine to recognize. So to do this, to train SVM classifier, we use 80% of the data set for training, and we keep other 20% for testing. And then we do tenfold cross-validation, and then we generate, machine generate best parameter to determine. And then using the other 20% to machine test, and then it provides accuracy. It's very simple, very straightforward mechanisms of understanding. Then machine gives us a decision function. This is the value, always machine gives us. Then we decide, or we use this information to decide where the samples belong to. This is the data. Out of 2,000 genomes, 17 guys are isolated from humans, 17 guys belong to animals. And 10 out of almost 300 belong to humans. Okay? So accuracy level is almost 98.9%. Compared to phylogenetic trees I used using MLSD data, the accuracy was about 50%. Maybe about 70%. About 30% of the genome was mispredicted, as also they are interspersed with other sources. So accuracy was significantly improved. And these guys were mispredicted. However, when we look at individual genomes, we found that these guys were isolated from farm workers, slaughterhouse workers. Therefore, still there is a possibility that this massa was actually originated from humans. Animals, not from humans. These genomes also mispredicted as humans because it was isolated from farms. It could be human massa, but it went to the animals. It's truly possible. But still, even if I delete these guys, I believe the accuracy level is pretty high. So then we used this model we developed to test new set of data. We isolated 62 more massa strains from human patients, exchange hospitals, and then we sequenced newly isolated from animals. All of them were predicted correctly about the origin of the source. Therefore, we believe machine learning gives us very accurate information. So now I can say very confidently, we have a power to discriminate where did they come from. Then your question will be, so what, right? And then, or if you have even better knowledge of the machine learning, then you're going to add, or microbiology, you're going to say, how machine learning could differentiate? What is the factor machine use to differentiate? Therefore, we used this analysis, permutation analysis. So it's very complicated, but at the end, what we found was these genes are really important that machine used to discriminate. And if the numbers are high, that means they are more frequently or more heavily used to determine the entire genome's origin. Even though there are more than 2,000 genes in MASA, we identified these group of genes. They were very specifically used by machine to understand. And right here, XERC4, XER5, those are the tyrosine recombinase. But here was another one, XERC. All of the XERC genes were very important that machine used to differentiate. And I look at the XERC gene prevalence as here, animal and humans. Animal and humans were carrying XERC4, XERC5, but most of human isolators only carried XERC. So XERC is very important to discriminate. Then we look at the XERC genes, and we found the XERC3 was encoded in phage genomes. And in phage genomes, they also carried other virulence factors like this. And then this is the XERC loci in the prophage 5 and 3 on 15. Most of the 5 and 3 on 5 was isolated from human genomes, not in animal genomes. And we found the XERC 5 and 3 on 15 is inserted in the middle of the HLB genes. And then once we treated this bacteria, we did all of the antibiotic treatment test to see whether this phage is prophage or not. So prophage means once they are exposed to the antibiotics, they're going to go to the lytic cycle and they kill the bacteria. So a lot of bacteria were killed by adding antibiotics like this. But some of them, like this strain, once they die, but they could recover to the normal levels. So we found 3 different types of phages. And this is different plasmid, different phage, but I would like to skip. And more importantly, these phages, they carried many different types of virulence factors, like here. Sometimes they carried the superantigen enterotoxins like this. Therefore, we thought maybe if we induce phages with antibiotics, maybe the virulence goes up because they now have intact HLB gene, which is hemolysin. Hemolysin, everybody knows, I believe, disrupts the cell membranes and causes disease. And we look at the hemolysin gene copy numbers once we activated the phages, activated the phages by adding antibiotics. And then we measured the cytotoxicity. When they have phage genes, the virulence went up significantly. Again, also we look at other virulence factors like this. When the phage was exposed to, this bacteria was exposed to antibiotics, the virulence even went up. It suggests a lot of information because if we use antibiotics to cure MRSA, it may even make it worse because the pathogenicity of the MRSA strains goes up, especially if they are specialized in humans. So here is my summary of lesson 3. So machine learning enabled analysis shows MRSA is a specialist. Most of the MRSA strains isolated are separated between humans and animals, suggesting we may not see frequent jumping between 2 different hosts. Even though we isolate MRSA from meat or animals, they may not transfer to humans. And phages play key roles in host adaptation. I'm not sure whether it is good or bad, but I believe it is good. And here's my summary. And MRSA is a global concern. There's no question about that. AMR spread happens everywhere. But even if a spillover event happens, they may not colonize in different hosts at this moment. Yes, many bacteria still, they have a capability to adapt to to different hosts. That's why we have learned, that's why we believe so far. But now, my question is, are we safe now from AMR in general? Okay. Maybe, I need to visit about the spillover. Spillover event means bacteria from MRSA from humans, or MRSA from animals may come to me, and one time they can be isolated from my body. However, they cannot colonize. Our immune systems, we block it. Therefore, they can colonize it, therefore they cannot cause disease. That is the definition of spillover and colonization. So with that, I'd like to acknowledge, Yu Ting-zai. She has done most of the work I presented today. And please do not share this information. This work is not published yet. Last night, as I said again, I just changed the second chapters completely because I don't want to present over and over already published data that you can read it from my papers. Okay? I think that's it.
Thank you, Casey, again, for excellent presentation. So we open to questions. Anybody?
Thank you, Professor, for your outstanding presentation and this focus on MRSA. So the question, mainly for the student, what exactly you can suggest beside antibiotics? Is there any other options to treat this harmful bacteria? And another maybe secondary question, it could be on MRSA from the nostrils. Is it becoming a problem, or what? You have to comment on that. Thank you.
Yeah, to answer to your first question, yes, is there alternative ways to prevent bacterial colonization or bacterial disease without using antibiotics? Of course, we have. So you're going to present the probiotics, yes. So probiotics is the, I believe that's the solution. Also enhancing our immunity, that is also the best one. And then second question, the MRSA is prevail. So MRSA, the prevalence of MRSA declining. Because when we have President Obama back in 2009 or 8, I'm not sure. So at the time, he made an executive order to fight against AMR. He defined AMR as a threat for national security. After that, we started to control. We built a task force to work on AMR reduction. I believe MRSA started to decline at that time, from that time. So, okay.
Any more questions?
Paul has, yeah.
Great stuff again, Casey, particularly the last new stuff. So I wasn't quite sure what data you were showing with those slides at the end. So if you introduce your phage genes, or have you introduced your phage genes into strains which don't normally have them? I assume you have, but you can assuage me here as well. And then do you get the phenotypes that you're talking about? So that's number one. And secondly, there are things like, you'll be aware of the CRISPR-Cas. Can you get, all that stuff in to delete this? Is that an option for therapy, or is there another way that you can think of that you might be able, if it's so important, that you can activate it? And third, we need to talk about another species.
Okay, good. Thank you very much. Second question first. So we reached at this point about almost a year ago. The power of machine learning speeded it up to generate hypotheses. But we did a lot of in vitro assays. And at the end, to confirm the information, yes, we definitely need to delete these genes. And we used different deletion methods, but we failed because this is MARSA, it's so hard to do transform. And we used, we end up using CRISPR-Cas9. Yeah, so now we are deleting it. Once we delete, maybe I can answer whether we can use CRISPR-Cas9 as a treatment purpose. And then first question, I'm not sure quite well. What was your main question?
You introduce plasmids in which CRISPR, Cas, and all that kind of stuff to delete specific genes. So if this is really is so important to that species, then actually that might be an approach that could be used so long down the line.
Yeah.
But that's a long way down the line. So I wondered if you'd thought about, I don't know, but other ways that you could actually inactivate this, phage genes, which would make a big difference if its effect is virulence, antivirulence therapy, essentially.
Sure. Yeah. Right now, I don't have any clue how to do it, to be honest. Maybe you can suggest some ideas after this seminar. Okay. Thank you.
We have time yet. Any more questions?
After Amin, I'm not going to answer to professors' questions, except the students. So students, please ask any questions.
But
you can say,
students can
Dr. John,
still-
you look so handsome. I'm fine to hear that.
Students can still get the priority, so I'm going to stop.
We have time, don't worry.
Hi, thank you. Now I'm shy because I'm a student, okay? So you can answer.
Okay, yeah, okay.
I have a question related to these markers that you find with the whole genome and the machine learning. Do you think that's the way that we're going to find, like, where the resistance come from in the future?
I believe so. That's the way I think we should move forward. Yeah, the answer is yes.
Yes.
I believe that is the case. That's the reason why I'm trying to validate what we found from machine learning by using a genetic method to test. If we fail to prove these genes are really important for colonization in humans, I'm going to throw away all of the machine learning analysis.
Yeah, maybe in the future.
Yes.
Thank you.
More questions? So I will go back to Amin.
Thank you very much, Casey. It was really nice presentation, and I have learned a lot, and of course about the machine learning. The thing is that my question is to some extent related to Raphael. So the thing is that the signature genes that you are going to identify with machine learning, and then definitely the machine learning approach will not be used by many of the people. So if you, let's say, develop something, whether that approach can be applicable to the other labs, whether they can use those signature genes to identify the transmission of AMR, let's say, in a different context.
Of course. Let me share some of those slides. I skipped. So, I believe you are using this information, these technologies. So, a lot of people, if you work in advanced labs, we use whole genome DNA sequencing to understand genetic markers. They may change the pattern of colonization. Before, like this MARSA, they were not pathogenic. In these chickens and humans, farmers, they are well adapted to these hosts. That means well adapted means they do not cause disease. However, they acquired some genes This is CC5 in European countries, and then becoming a global spread, and they cause disease. Also, they go to the chickens now. Now they cause also disease in chickens by adding one or two genes. This is what we have done so far, last 10 years, maybe 20 years. Once we have the first E. coli genomes sequenced, everybody started to look at genes. They are responsible for enhanced virulence, enhanced pathogenicity. But how many years it took to get this information? Once they sequence them, they have to compare one by one to identify the genes, and then we delete, and then we confirm in animals. It took about, if you work in very advanced labs, maybe it takes about 2 years or 3 years, including in vivo tests. But if you use, it can be done in maybe 10 days. And to get the hypothesis, this gene is important. Then we can set up another in vivo test to confirm. So I strongly urge you guys, students, to learn how to use machine learning and how to use it, because it provides you a hypothesis to all of the questions you may have. That speed up your work. So Yu Tingjia she's my PhD student. She came from food science. She was a food engineering student during her bachelor's degree. She has wine tasting certification because of her training. She was an engineer, but she came to my lab and she learned everything, microbiology, machine learning, within 4 years, and she generated this amount of data. Before, I had one time more than 10 students in my lab to work on each project. But now, over 2 or 3 people can do all of the work that we have achieved in the last 10 years. So it speeds up a lot of your work in progress. It saves a lot of time and money. Therefore, I believe everybody should use. I hope that's the answer to your question.
So, Casey, we have a question here from Rosvir.
Okay.
She's asking, what is your opinion about...
What is my what?
Your opinion.
European?
Your opinion.
My opinion.
Yeah, about farms that they don't use antibiotics.
Okay. That's really good questions. Actually, I had that chapter on my presentation today, but I decided to delete because it's already published. Now we have better understanding of natural resistance. Antibiotic genes are existing already in bacteria. So many times when I give a lecture about this, I say that if you go to the Amazon forest, the area has never been exposed to human activities. If you isolate some bacteria, they carry antibiotic resistance genes because bacteria, they produce, they generate these antibiotic resistance mechanisms to fight against other microorganisms, mainly some actinomyces. Flemings from Imperial College of London, he found from actinomyces. What's the name of the strain he found first isolated ampicillin? Okay. So anyway, so it was 1926. Am I right? 28. Okay. So it was 28, and we started to use antibiotics. From that time, more than 98 years, almost 100 years. But before that, bacteria, they started to use, bacteria, they use secreted antibiotics. Therefore, bacteria, they have mechanisms to resist against those antibiotics. Yes.
We have one more question here.
So, Professor, how do you think we can overcome the problem with resistance? We know nowadays that the mobile genetic elements are the main source of those genes, and they spread very quickly, and the more they spread, the more we use antibiotics. And how do you think we can reduce this spread or even mitigate the natural course of evolution with those genes?
Okay. that question looks like very sophisticated. But if I share this story, maybe you may have different opinion now. People, many people, they tend to accept the IS element. They have no barrier, and they can freely, jump to other genomes, other bacteria. Looks like they are on the highway. They can go to anywhere. However, when they are inserted into the certain genomes, they have a hotspot. This is the site. Normally, this IS element jump into these genes, CAPI genes. CAPI gene is now deleted. Also, even transposons, they have a hotspot for transposon insertion site. So even though we believe IS element, they can freely insert it to others, that's not true. If that is the case, all antibacterial plasmid, they can freely transmit or transform to other bacteria, IS element conjugation. We have at least antibacterial resistance for last 100 years. At this time, it must be exponential phase. But I feel like still we are at the lag phase because of this restriction. Many plasmid, they don't have, also many bacteria, they don't have natural transformation efficiency. Only few bacteria, including Acinetobacter or Legionella, they have natural transformation efficacy. So even though I have plasmid DNA here, there is a very low odds that this plasmid DNA can be transformed into my microbiota. That's my opinion. It could be wrong. Maybe Paul, you may have different opinion. Okay.
So I think some of the IS elements, like we use Mariner, you only need 18 to insert.
Sure.
So, that's in every genome. So I think, so it depends on the IS element, but I kind of agree with you that, well, it depends. So it just depends. I can't remember the second point you made, but I disagreed with something there as well. But
okay.
We can discuss.
Good. Yeah, that's the reason why I presented this work. I wanted to have open discussion before I publish. I submit and get rejected.
More questions? No?
Okay.
I think we're done. Okay,
Thank you.
so we will do a short break now, and we will return here for the next class. Okay, thank you so much. Thank you, Chris. So let's continue with the lectures. So our next guest is Dr. Amin. He's from Washington State University, and he's a microbiologist. And he will talk a little bit more about AMR. So thank you for coming here, Amin. And yeah, the stage is yours. Okay.
Thank you very much, Ricardo. So, good morning, everybody. First of all, I would like to thank the organizers, especially Nero, who invited me to attend this wonderful symposium. And for me, this is the first time in Brazil. And so far, I have been, enjoying every moment of it. And I have met so many wonderful people here and learned so many things, especially with the colleagues and, the guest speakers from other countries, especially, from here and the US and the European countries. So it's a great experience, learning experience for me. So my talk will be on AMR, but as I was saying, after Casey's talk, I was thinking that, okay, my talk should have been in the earlier because I will be asking some questions, and his presentation has already to some extent answered some of my questions. So I could have given a kind of introduction and asking questions, and then later on he could have jumped in. But it's okay. And I will be giving a kind of very, short overview of a kind of classic molecular epidemiological studies that we try to design to identify the sources of foodborne pathogens or sources of organisms that cause infections in humans. So in my case, I will be talking about the source attribution, especially the foodborne sources of E. coli, ESBL E. coli. By the time you already know what is ESBL, extended spectrum beta-lactamase, and this gene basically confers resistance to third and fourth generation cephalosporin antibiotics, and which are most commonly used for treatment of the gram-negative bacterial infection. So, and the health conditions or the disease that I will be mostly focusing on is the UTI, and this is community-acquired UTI, urinary tract infection. My name is Muhammad Aminul Islam, so this is a big name, but everybody calls me Amin, and I am with the Washington State University at the Department of Allen School for Global Health. So just before going into my presentation, I would like to give you, an overview of the location from where I am and what is Global Health Department at the WSU is doing. So the Washington State University is here, and when people they hear about Washington State University, they think that it's in Washington, D.C., but we are not in Washington, D.C. So it's in the Washington State. It's the west coast of the United States. It's just opposite of Washington, D.C. And since we are global health, so we are working in different places across the world to solve the global public health problem. And of course, antimicrobial resistance, as you have heard from previous speaker, is definitely a global public health problem. So we have the Allen School for Global Health. They have very well-established research programs in different places, including East Africa, Kenya, Tanzania, South America, in Argentina, Peru, Guatemala, and South and Southeast Asia, like Vietnam, Thailand, and Bangladesh. So I am originally from Bangladesh, and I have worked many years in Bangladesh with an organization I will talk you in a minute. And so I have a very close connection, and I have a very well-established field site over there that we can use to answer some of the questions that we ask and we raise often. So this is the organization. It's named ICDDRB. Previously, it was named as International Center for Diarrheal Disease Research. This institution is located in Dhaka, which is the capital of Bangladesh. And this institution is known, fairly known as a the premier research organization conducting research on diarrhea, dysentery, and enteric diseases and cholera. And now they have expanded their horizons to other non-communicable diseases research. So they have very good field sites at the population level. Also, they maintain a large enteric diseases hospital where they treat, I think, the thousands of patients with enteric diseases every year. So it's a very good setup. You have like the hospital settings where you have the exposure or the access to the patients, and you have DSS surveillance sites, demographic surveillance sites at the population level in the community, and these two things are also sometimes integrated. So it's a kind of, the nice setting to design any study that if you want to. And so in my case, why I am, doing this study on AMR in Bangladesh is not like only that I'm from Bangladesh, but Bangladesh is a kind of, or not the Bangladesh, but whole Indian subcontinent is a kind of hotspot for AMR, because, Casey was talking about New Delhi metallo-beta-lactamase, right? So New Delhi, so this enzyme was discovered and named after New Delhi, which is the capital of India. So the thing is that the use and misuse of antibiotics in those regions is pretty high. And at the same time, the infectious diseases caused by antimicrobial resistant organisms is also pretty high. So altogether, this makes a kind of very conducive situation and the population to study antimicrobial resistance. And to some extent, if you have a focus on the epidemiology or the population level studies, I think these are very good environment to provide you the support. So this is a, this information you have seen many times, I think, and Casey has also showed some of the statistics here, but he was mentioning about the, the statistics in the United States, but I will be, broaden it a little bit to the global scale. And as you know, this is, okay. So this one is a kind of assumption, but this one is from a recent study which was published in Lancet. And they reported that only in the year 2019, approximately 5 million deaths were associated with AMR infections. And of these 5 million deaths, 1.27 million deaths were attributable to bacterial infection, which are antimicrobial resistance. So globally, I think the burden of AMR infection, the death caused by AMR infection is pretty high. But the thing is that if we don't take any actions, and if we don't put any interventions or do anything right now, this number can raise up to 10 million per year by the year 2050. So this is a very big number, and of course, this will incur a huge economic cost. And obviously, the low and middle income countries will be disproportionately impacted because they have the bigger problem to solve, but they have low resources and low capacity to deal with this problem. Now, apart from these huge health burdens and the economic burdens, it is also assumed, or maybe it's already happening right now, that AMR will be heavily impacted the attainment of sustainable development goals. I know that, by the time you already have idea about the sustainable development goals that has been, set by the UN. And at least 5 out of 17 SDGs, Sustainable Development Goals, will be directly impacted by AMR. And these are like the no poverty, zero hunger, good health, well-being, reduced inequality, decent work, and economic growth. So basically, without addressing the AMR problem, it will be really difficult to achieve the goal for sustainable development that has been put through. So, considering these huge consequences, the World Health Organization has recommended to include AMR-related indicators to measure the Sustainable Development Goals or the progress of the Sustainable Development Goals. And specifically, they recommended to track two most important drug-resistant pathogens. One is the E. coli resistant to third-generation cephalosporin, and the other one is methicillin-resistant Staphylococcus aureus. So, Dr. Casey has already discussed a lot about the methicillin-resistant or MRSA, and he also touched upon a little bit on the E. coli that are resistant to third-generation cephalosporin antibiotics. So my talk will be focused on the E. coli resistant to third-generation cephalosporin. And so these are also, like we call it, ESBL producing, because the ESBL is the enzyme that causes or that confers the resistance to third and fourth generation cephalosporin. So I will be using the term interchangeably, ESBL, E. coli, or 3GC resistant E. coli. So a recent study or recent meta-analysis with the data from almost like 49 different countries, they found that the E. coli that are causing bloodstream infections globally, 36% of them are third-generation cephalosporin resistant. So the burden is pretty huge. So apart from these, the bloodstream infections and the diseases, the other compounding concern is that the colonization of healthy individuals with these organisms. So the colonization of ESBL E. coli has also been increasing globally. And a recent meta-analysis has shown that the colonization with ESBL E. coli also varies from the region to region, population to population, with the highest has been shown in South Asia region, accounting for approximately 25% of the people. So the 25% of the people across the board are colonized with ESBL producing E. coli, which is a very, the high prevalence, right? So I have been studying, I have done several studies in Bangladesh to estimate the colonization with ESBL E. coli in different, the population groups, in different age groups in Bangladesh. And what we have found in a study with children less than 1 year of age, we found that almost like 78% of the children are colonized with ESBL producing E. coli. So think about the global estimate and the estimate in Bangladesh. So, and when we look at this is the distribution of ESBL E. coli prevalence according to month. So when we looked at the age groups, we found that the infants, or let's say the children, even with 1 year of age, they are heavily being colonized with ESBL producing E. coli. And we have done another study, I haven't shown it here, but we collected the rectal swab samples from infants within 48 hours, and we also found that those infants are being colonized with ESBL producing E. coli. And we try to count the number of ESBL E. coli in stool samples from these kids, and we found that sometimes the number is so high, like 10 to the power 5 CFU per gram of stool. So it's very scary. But yeah, we will talk about this. And then among the adult population in a different study, we found that across the age groups in the adult population, on an average, 75% of people were colonized by these ESBL producing E. coli in Bangladesh. So this is also very high. And one of the objectives of this study was to look at whether there is a kind of risk of ESBL E. coli colonization among people who have regular exposure to poultry, especially the farm poultry, compared to people who do not have. And we found that the people having occupational exposure to poultry have a higher risk of colonization or higher prevalence of colonization with ESBL E. coli. We also found some differences between the people living in urban areas and rural areas, but that difference was not statistically significant. So now the question is, do we really need to care about that? So this, the high level of colonization with ESBL, Enterobacteriaceae or ESBL E. coli, do you need to worry about that? So some of the, the things Dr. Casey has discussed already, but I want to say that yes, we need to care about that ESBL E. coli colonization, because studies have shown that, and we also have found in our study that the people with high level of colonization with these organisms are at higher risk of being infected by the same organism. And most of these studies, the problem is that most of these studies have been done in the hospital settings, in the hospitalized patients. Because the studies was like that, the people, they get admitted to the hospital, and before getting the admission, their samples were collected and screened for colonization with those organisms, and then they were followed up during the hospitalization, during the hospital stay to see, what proportion of them developed the infection and the etiology of the infection. And there is a clear link that the people with prior colonization from the community are definitely at higher risk of infected by the same organisms. But we are talking about the community, we are talking about the population level. And it is hard to get this information because you need to do a prospective study and follow-up study, and those are very expensive. Those studies haven't been done. I haven't seen so many studies, but I will give you an example. This was a nice example that I got, and this could be, the setup, some of the premises of why we need to care about this. So this study was, it's a large population-based study conducted in Sweden a few years back, where they have used national health registry data. So they enrolled like almost 23,000 people. So 23,000 people, of these, 17,000 people had the first reported diagnosis of, or first detection of ESBL E. coli in their urine sample. And then almost like 5,000, more than 5,000 had the first reported diagnosis of E. coli in their feces. And these people, the group of people, were being followed up for 6 years. And in addition to that, they also included more than 45,000 people who do not have any reported diagnosis of E. coli either in their urine or in the feces. So it's a classic case control study. So they followed up these people for 6 years, and the major outcome measures that they were looking in this study was bloodstream infection caused by ESBL-E. And what they have found, the cumulative incidence, 6-year cumulative incidence of bloodstream infection with ESBL-E was 3.8% for people who were colonized in urine, and almost like 2%, I think 1.8% precisely, for the people who had colonization in the feces or intestinal colonization. So which means that we need to care about that. And if you see the comparison group, they do not have any bloodstream infection. So it says that, or it tells us that yes, we should care about that, and we should take steps, measures, or interventions to reduce the burden of colonization at the community level, at the population level. Now, what interesting thing they have also found here is that almost 87% of the cases, they found that the same species is causing the infection. So like E. coli in the baseline, and then E. coli. So they were working on ESBL-E, Enterobacteriaceae. So they found the same species is causing the infection. So they found in the baseline E. coli, and they also found the E. coli is causing the infection, bloodstream infection. And 80% of the cases, it was E. coli. So definitely we need to care about that. Now, apart from the bloodstream infection, there are other, the extra intestinal infections that we need to think about or we need to consider that are being caused by extra intestinal pathogenic E. coli. So if you are not familiar with the term extra intestinal pathogenic E. coli, just to let you know that we can broadly categorize E. coli into pathogenic and non-pathogenic, right? Non-pathogenic are, to some extent, the commensal organisms resides in our gut. But the pathogenic E. coli can be classified into two groups. Simply XPEC, extra-intestinal pathogenic E. coli, and IPEC is intestinal pathogenic E. coli. So as a food safety specialist or food safety or microbiologist, we are quite familiar with the term intestinal pathogenic E. coli because that's what we have been dealing with, right? And we know the classification of the E. coli under this, like enterotoxigenic E. coli, pathogenic E. coli, enteroinvasive E. coli, diffuse adherent E. coli, hemorrhagic E. coli or sugar toxin producing E. coli, and aggregative E. coli. But then we have this XPEC, which is like depending on the site of infection, where these organisms cause infection, they are classified into different types. Like the E. coli causing infection in urinary tract, UPEC. And then NMEC is causing the neonatal meningitis. Then MPEC is the pathogenic E. coli that are causing infection in mammary glands. And then SEPEC, which is like the septicemia-associated pathogenic E. coli. So the other thing is that a group of XPEC is also causing extra-intestinal infections in poultry, for example. Poultry or birds and pets and animals that are named as APEC, avian pathogenic E. coli. So now I will be moving to some extent to the UTI or the community-acquired UTI, and is the UTI caused by multi-drug resistant organisms is a significant public health problem. And in the United States alone, approximately 8 million medical visits, which including 1 million emergency room visits and 100,000 cases of hospitalization occurs every year, which incurs like the more than $1 billion US dollar per year. And of course, E. coli is the predominant cause because of its multi-drug resistant properties. Now, what we have discussed in the previous slides, that when these organisms are in our gut, they usually do not make any problem. So because these are extra-intestinal pathogenic E. coli, they don't express their pathogenic properties or the virulence properties while they are in the gut. But it is also conceivable that somehow these organisms gets into the urethra and then transcend to the urinary bladder where they colonize and cause infection. And then in more severe cases, it transcends to kidney, bloodstream, and then go to other organs where they can cause more severe infections. Now the problem is that when these organisms in the gut, they don't make any problem, and then we don't know, when it will cause infection after colonization. So the time lapses between the colonization and infection varies. And that's why, it is really difficult to identify these organisms while they are colonizing in the gut. And the virulence genes or the factors that are being used to identify the UPEC strains, those when they are in the gut, they are more or less common with the other commensal organisms. So there is even not a very good system, not a very good identification markers that you can use to identify them in the gut, and so that you can monitor. But even, I think, you find the organisms in the gut, but you don't know whether that will cause any extraintestinal infection or not. So that makes really, the source attribution very, very difficult and challenging in this case. So considering this huge problem, there have been a lot of efforts to find out the sources of urinary pathogenic E. coli. And the most important sources, or most important, the areas that had been studied so far is the poultry, and the poultry meat contamination with the uropathogenic E. coli. And the premise for this is, that I mentioned, is, the avian pathogenic E. coli. It has been shown that these avian pathogenic E. coli are genetically related to E. coli or uropathogenic E. coli. But the problem with these, these studies, is that the organisms or the samples were collected at, the different time and different places. So patient samples were compared with, the food samples or the meat samples, the isolates from the meat samples at different time and space, with no or little epidemiological collections, and that's why the source attribution is not accurate or is not, the clear. So what we have done in our study, we have designed a study where we wanted to, address this issue, and we designed this study in such a way so that we can collect the patient samples and the food samples at the same time and space, so that we can have the epidemiological connection, and then we do the phylogenetic analysis to find out the relatedness of these organisms and whether there is any sharing between the isolates from different sources. And we have done this study in Bangladesh, in Dhaka. And as I mentioned, the collaborating institute is ICD-DRB. So this is the map of Dhaka, and this is the location of ICD-DRB. So ICD-DRB, as I mentioned earlier, they run a very big diagnostic clinics where the people from the community, so they come and then they can test their clinical specimens. So over the period of 2016 and 2018, we enrolled like 3,200 patients who attended this ICD-DRB diagnostic clinic to test their urine samples for culture and sensitivity. So we tested all these urine samples following the standard clinical microbiology guideline. And of these, 3,200 patients, we found that like 920, almost like 30% of the patients had significant bacterial growth of a single bacterium, like 10 to the power 5 CFU per ml of urine, which indicates the laboratory confirmed cases of UTI. So if you have, the clean catch urine sample and you have growth of a single bacterium with a threshold of 10 to the power 5 CFU per ml, then we usually, identify that as a possible case of UTI, provided that you have like symptoms. So all these patients were having like suspected cases of UTI. They were attending the clinic to test their urine samples whether they have any sort of the urinary tract infections. So we enrolled them in our study, and then we tested their urine samples. And of these, 30% of the patients that were positive for UTI, community-acquired UTI, because these are not hospitalized patients, these were from the community. So 52% of the patients were positive for E. coli, as we confirmed by Vitek2 system. And then we carried out the antibiotic susceptibility testing of all these isolates using sensititer plate, and we confirmed their ESBL phenotype by using combined disk test. So we found like 55% of these E. coli were ESBL producing. And at the same time, we collected like different types of food samples from 23 wet markets. I don't know how to explain the wet markets. Probably you also have wet markets here. But we have two different kinds of markets. One is the retail, the two different categories of retail market. One is the wet market and the other one is the super shop. So these wet markets are pretty popular and most of the people go to the wet markets to get their groceries. So we have collected different types of samples from 23 wet markets, which is located within the 10 miles radius of the ICD Derby Clinic to have some epidemiological connections. So the samples that we have collected from these markets was poultry meat samples, which includes broiler chicken, then the free-range chicken, pigeon, duck, etc. Then we also collected the beef samples, the raw beef samples, and the fish samples raw fish samples of two different, the most commonly consumed fish types, tilapia, and one of the catfish, I don't know its name, as pangash. And then we also collected raw egg samples, including chicken and duck eggs, and the fresh produce samples, including carrots, potato, and spinach. So we sampled every market once in a month from these 23 markets, and a total of 2,297 samples were collected over a period of 1 year. Then we processed all the samples following a standard guideline. In this case, we followed the ISO guideline to process all these food samples, and then we used the chrome agar ESBL plate to identify the ESBL producing E. coli, and then we confirmed the E. coli using the VITEC and their ESBL phenotype using the CDT, the combination disk test, and we found that almost like 30%, I think it's 28% precisely, 28% of the food samples was positive for ESBL producing E. coli. Then we tested all these organisms, all these ESBL producing E. coli from both from food and clinical sources, E. coli phylotyping by PCR, and we took representative of the isolates from each of the phylotypes. In most cases, the overlapping phylotypes. The overlapping phylotypes means overlapping between the human and the food sources, and used the whole genome sequencing to do phylogenetic analysis. So now, a little bit about the results. So as you can see here, the prevalence of ESBL producing E. coli in food samples, in case of broiler meat, it was the predominant, almost 55% of the meat samples. Broiler meat samples was positive for ESBL E. coli, followed by duck meat and the free-range chicken, beef. So, but compared to the fish, meat samples, the prevalence of ESBL E. coli in fresh produce was relatively low, but still very high when we compare this data with the global estimate. When we looked at the antibiotic susceptibility pattern of these isolates and compare between the food and clinical sources, we found that the clinical isolates were predominantly resistant to many different antibiotics, including third-generation cephalosporin, of course, and then fluoroquinolones, aztreonam. But surprisingly, we found that the food isolates, they were predominantly resistant to some antibiotics like polymyxin, colistin, and tetracycline somewhere. So this is the doxycycline, tetracycline. So we haven't dig deep into why is the cause, but probably the cause is that these antibiotics are being heavily used in raising, the chicken or in the poultry farms. But we haven't collected any information from the farm, so we cannot comment on that. But it is conceivable, and also from Casey's presentation, that the colistin are being heavily used in the poultry farms across the world. So that could be one of the reasons. Now, when we looked at the phylo groupings of these ESBL E. coli, it was quite interesting. You see the clustering based on the phylo groups. The clinical isolates, almost like 63% of the isolates were clustered in phylo group B2. Interestingly, none of these, the food isolates belong to this phylo group. On the other hand, you see the 52% of the food isolates belong to phylo group A, but only 4% of the clinical isolates belong to this phylo group. So this was kind of interesting in the beginning. So they have a kind of clear separation, and clustering based on their phylo group. Phylo group is a very preliminary, classification methods that we can use. But even they were clustered separate. And the overlapping phylo groups were like D, C, F, but the proportion of the isolates in each of these phylo groups was very small. Now, when we looked into the whole genome sequencing and we did categorized them based on their MLST, the multilocus sequence typing of these isolates, we found in case of the clinical isolates, it's interesting, it also matches with the phylo grouping results that I have just shown. Among these 111 isolates, we found 23 ST, but 56% of these isolates belong to a single ST type, which is ST131. And you know that this ST131 is a pandemic clone or pandemic type, which is one of the major causes of extraintestinal infections globally, including UTI. So I was not expecting these results that this will be so clustered and this ST131 will be so prevalent in the community. So when we looked at the distribution of ST types among the food isolates, we found it's all diverse. And we didn't see any sort of clustering based on any specific ST types. Like with phylo grouping, we saw some, more than 50% of the isolates belong to phylo group A, but here the ST was all dispersed. So of 102 isolates that we tested, we identified almost like 50 ST and no clustering. So when we looked into the ST types that were overlapping between the food and clinical isolates, we found only 4 of these, ST648, 38, 10, and 457. So there are multiple strains belonging to each of these ST types that were overlapping between human and poultry meat isolates. So we looked into the specific O and H type or the serotypes of these isolates. Each of these isolates, we then found 3 ST types had the overlapping isolates having the overlapping serotypes. When we looked into the SNP differences, a single nucleotide polymorphism between these isolates, we found that SNP differences is between like 30 to 73 among these overlapping isolates. And in our case, as for determining the clonal identity or clonal relationship, we considered a threshold of 20 or less than 20 SNP differences based on some previous epidemiological information. And according to that threshold, we don't say these are clonal. Maybe they have originated from the same ancestral clone, but they were not clonal. So this is the core genome phylogeny, the maximum likelihood phylogenetic tree. I don't think you can see it very clearly, but these, the shaded isolates are basically the isolates that we found clonal to each other, so less than 20, 20 or less than 20. But most of these are between, the isolates either within human or within the food sources. So there was no overlapping between the humans and clinical or food sources. So since the ST131 was quite prevalent, so we wanted to do some more characterization of these isolates to see what sort of properties they have. And we found very interesting is that almost like 85% of these ST131 belong to clade C, which is very, very important because they are also fluoroquinolone resistant, and also they carry the BLA CTXM15 gene. And they are positive for FIMH30, which is the signature gene for the classifying ST131 into C clades, especially the C2 clades. Now, what I have shown is basically, doing the core genome sequence analysis and trying to find out the clonal relationship. But since we didn't find any clonal relationship between isolates from different sources, we thought that probably mobile genetic elements or the accessory genes could be something that transfers the resistant genes, and that could be one of the important aspects of the transmission of AMR. So we started looking at mobile genetic elements, and this is a very busy and comprehensive slide, but just to show you, and these are not all the MGs that we found is the most, the top, I think 60 or 70 different MGs that we try to cluster them together. And these are the humans. This is chicken, duck, and pigeon. As you can see, some of these MGs are commonly present across different sources, but some of these MGs are also, the unique to different sources. So we were planning to do the long-read sequencing of these isolates, some of these interesting isolates, to extract the plasmid, and so that we can characterize this plasmid and to identify whether these plasmids carry antibiotic-resistant genes and those have any epidemiological potential. So while we are doing this, and so let's, I will skip this conclusion right now. While we were planning on this, and we found this paper came up. This paper was published by Lance Price from George Washington University. And they basically, reported that they have identified some source-associated mobile genetic elements where they can use to identify the zoonotic transmission of E. coli infections. So what they have done, they had a kind of similar, the study design that we have done. So they also collected the clinical samples and food samples contemporaneously or concurrently with epidemiological, the connections to identify the foodborne sources of UTI. But they had, they tested way more than isolates that we had. So they basically did whole genome sequencing of 3,120 isolates of these, almost 2,000 meat isolates, 1,100 human clinical blood and urine isolates. What they have found, they identified total of 443 ST types. Of these, 120 was human and 247 in meat, and they also found 76 STs was overlapping between the two sources. So similar to what we have found, but we found in a smaller scale with the same study design. But when they looked into the SNP differences, and they also had the same conclusions that they didn't have any sort of strains overlapping or transition from food sources to the human sources. Casey is smiling because that's what I was trying to say. So they have used so many strains and did whole genome sequencing of. So we were thinking, okay, we have a smaller number of samples or strains. That could be a kind of, in terms of, let's say, the power of the studies or everything could be some drawbacks or setbacks. But they have done also, they address this problem, and they have included so many strains, and they also came up with the same sort of conclusion. But what they have done, they took a different path, and they extended this study, and they go for the accessory genome analysis. So they try to find out the accessory genomes or accessory genes which are differentially present in isolates. So they compared they did the pairwise comparison among the isolates and try to find out the accessory genes that are differentially present, and then put those accessory genes into cluster or elements, then that they called mobile genetic elements. And using that approach, they identified 17 host-associated MGEs in human and meat isolates. And of these, 6 were human-specific and 11 were meat-specific. So it's not like, the only one gene, but these mobile genetic elements, this could be plasmid, this could be prophase, but they carries like between 2 to 42 accessory genes altogether, and they make sure that these are host-specific. So these are, the obtained from either, so the consistently present in any of the sources. Now, when they used these, the genes, the mobile genetic elements to identify the sources, you see the cumulative use of these, like all 6 genes, the sensitivity is very high. In case of, the meat-associated MGs, you see the sensitivity is more than 98%. And for the human cases, it is also like more than 90%. But the specificity is so low. So you cannot use this algorithm to identify the source-associated, the isolates, or you cannot predict that based on that. So to optimize this, ratio between the sensitivity and specificity, what they have done, they put everything, all these, the accessory genes and mobile genetic elements as a, the binary variable, and then put it in the Bayesian Latent class model, okay? Including their, the pan-ST grouping, the ST types that they have. So ST types in conjunction with their mobile genetic elements or the accessory genes, they use this latent class model, Bayesian latent class model to measure the probabilistic, of identification of the source-associated isolates. So like in this case, they used a threshold of 0.8, which is the probability that they considered. If the probability is 0.8 or more, then they will consider this as positive for source attribution, and if it is less than or equal to 0.2, is negative. So using that approach, they identified that 8% of the human isolates were basically predicted to be, according to their model, is from the meat sources, the chicken sources. Okay? So this was their finding based on the accessory genome analysis. But in case of meat samples, the probability of isolates from human origin was very low. So this basically provided a big hype, I think, when this paper was published, and this was covered by all, the big news media, that the UTI strains or the E. coli strains, multi-drug E. coli resistant strains, multi-drug resistant E. coli strains that are causing community-acquired UTI, 8% is coming from the food sources. So it was a big hype. So just to give you an example what they have done a little bit, so this is a classic example of ST48 isolates, and these are the, the sample types that they have used. Red is the meat, and then this olive color is the human sources. As you can see here, this one as an example, so this is although from the human sources, but the model says that it is more than 80% cases it is from the meat sources. So they use these, the classification of MGEs the type of MGs, the algorithm, and including their pan-ST classification. So just to summarize what I have presented here is, I would say that core genome-based sequence analysis of the isolates alone is not sufficient. It's not sufficient to identify the potential sources of E. coli causing UTI or any other extraintestinal infections. And I know that many of the laboratories across the world still, they don't have access to even the whole genome sequencing, but they are doing whole genome sequencing and trying to find out the epidemiological connections, or trying to find out the sources of the organisms that are causing clinical infections. But definitely there is a big caveat here. So WGS alone will not be able to attribute the source. And probably what I have here, like the mobile genetic elements, could be source specific. I don't know. That's why I asked the question to Casey, that whether, doing the machine learning things, if you could identify some signature genes that can be applicable to the other context, or maybe other set of samples, whether can we use those as signature genes to identify the sources. That could be something that we think about in the future. But definitely we need a good system, because whole genome sequencing alone is not sufficient. You need to add something. Like there they use like predictive models, but machine learning could be something that can be integrated. But how to integrate, and whether you have to do the machine learning by yourself, extracting the data from the online or from the website, or you can already use something which has been developed by somebody else. That is a big question. So I just want to finish it up here, and to acknowledge the people, the wonderful people at ICDDRB who did a lot of work collecting the samples, initial processing of the samples, sending the samples to WSU. And at WSU, my grad student and postdoc, so they did the analysis and so on. And I also like to acknowledge the funding agency, NIH, for providing this fund for this study. And specifically, I would like to mention here Dr. Lee Riley. I don't know if you know him. He's from UC Berkeley, and he was my co-investigator in this project, but he passed away 2022. So he was a great mentor and great collaborator in this project. And I thought that I should also pay tribute to his contribution to this project. Thank you so much. Thanks for patient hearing, and I'd like to have questions if you have.
Thank you, Amin, for excellent presentation. So we have some minutes and we can open for a question. Anyone has?
Thank you, Amil, for your presentation. I'm a very naive person regarding genomics. I'm learning. This is the reason I'm with Casey in Florida. But I wonder, this analysis you have done, for E. coli to produce an extraintestinal infection, they have to show some virulence genes, right? Are those virulence genes that supposed to be in those clinical isolates also in the meat isolates or food isolates? Did you check if those virulent genes were in both?
Yeah. We have checked it. I haven't showed it here, but we have checked. These virulent genes are commonly present. But the thing is that we make sure that the clinical isolates, basically, they need to be perfectly defined. So as I mentioned that UTI cases, it should be like 10 to the power 5 of a single organism, clean cats, urine samples, and there should be no contamination and have some sort of symptomatic connections. So that is the definition of classical UTI. So these are, since we've maintained all these definitions, these are all clinical isolates causing the UTI. And then when we looked at the virulence profiles of these isolates, comparing the ESBL, E. coli from food and clinical sources, we tested all these. But what I mentioned that those virulent genes, I mean, you have like, you will see the papers which are considering 20, 30, or 50 virulence marker, but those are not quite specific like as we used for the dirogenic E. coli. Like we have a specific marker for enterotoxigenic E. coli, like enteroaggregative E. coli. But for the identification of the extraintestinal pathogenic E. coli, we don't have those signature genes or markers that we can use for diagnostic purposes.
I have one question and one comment. First, my question is the Bangladesh data. So when you show you the prevalence of ESBLs in food sources, it's surprisingly or strikingly high. How could it be 50% from poultry, from chickens? Have you checked the gene prevalence of CTXM genes, OXA and others?
Yeah.
Do they have all of the CTXM genes?
Mostly CTXM15.
It's very surprising to me. In the US, we do not see that.
Yeah. Yeah, that's surprising, and we have also published many of these papers, about this high prevalence in. So we have, this is the food sources, but I basically work mostly on One Health. So I have like wastewater, surface water, the soil samples at the household environment. We have a pretty high prevalence of ESBL. That is surprising, and that's why, my concern is that whether we should care about that, whether we should be concerned about that, because ESBL is so widely prevalent in the environment. So regarding your questions, like why it's so high in the meat samples, so probably...
Can you show me your slide?
Yeah.
Yeah, that one.
Yeah, it's like 55% of the broiler meat samples. So it's probably, when we, this is meat samples, right? These are not fecal samples. But when we tested the fecal samples, individual, like cloaca samples from individual poultry, I believe, if I remember it correctly, we found that almost like 77% of these chicken were positive for ESBL E. coli in their cloaca samples, individual birds, not the poultry droppings or fecal. Yeah.
Even carrot and potato, they have high.
Yeah, so that's why I was telling you about a little bit of the wet markets. If you have any idea about the wet markets, if you have been to Southeast Asian countries, you see everything is under the same premises. So you have like large animal slaughterhouse, not the slaughterhouse, but the slaughter on premise. And then you have like poultry sheds where they are selling the poultry, slaughtering them, cleaning them. You have at the side fish, and then you have the fresh produce. So it's all together under the same shed. And people are, they going, and then when there is a rain, everything is, they mixing up together. So probably the fresh produce one could be like the cross-contamination, or we don't know, maybe it's coming from the field, because in the environment it is also predominant, it is also the heavily present.
Okay. Yeah, so good thing is there's no clonal variants isolated from food source and human clinical isolated. That's a good thing, right?
Yeah, so that's,
if we didn't have whole genome DNA sequencing techniques, if we just identified CTXM genes about 20 years ago, everybody will say there is a huge transmission between food source bacterial pathogens to humans, and then they always blame producers.
Yeah,
right?
I agree.
So that's the beauty of having these kind of advanced technologies. Now, my comment for Lance Price is, okay, they failed also to isolate clonal variants, the same isolated from food source versus clinical isolate. If there is a case, I think they had to stop there. They have to say there is no transmission between human and food sources, but rather than saying that, they decided to overfitting. And they look at the MGEs. Yes, MGE sequences are very small, relatively. Therefore, of course, there are always a similarity. Then they use similar information for source tracking.
Yeah.
One of the worst bioinformatics techniques is source tracking.
Tracking, yeah.
Therefore, I do not agree with their conclusion, and they shouldn't be, broadcasted and highlighted by a lot of news, because that is contaminating our opinion, and they are misleading to have bad policy.
So I totally agree with you, and I was really surprised because that's why, I put my conclusion before going to this Lance Prize thing, and I had the conclusion that there is no connection, okay? There is no overlapping, there is no zoonotic transmission. But in 23 November or October, they published it. And if you look into the website, you will see this news, because these are US population, right? Yeah. So the news was so big, and it was a hype. So it's like Wall Street Journal, The Guardian, the New York Times, everybody reported it, the 8%. So you get E. coli causing UTIs coming from the meat sources. So that's why I didn't know that he is doing this study, because it took us a long time to set up this kind of study at the population. It's not like we are extracting the data from website and did the analysis. We went to the field, we designed this study, and then we collected samples, we isolated the organisms, and we did the whole genome sequencing. It's a lot of efforts. So then when I see this, okay, I was happy that, okay, we are in agreement. But then they have used this, and it was a big hype. So I thought that maybe I should also try with these. And now maybe, we can also try with the machine learning things, because we have this data, how it works and whether. But to me, as the molecular epidemiologist, I think that we should end up here with the whole genome sequencing. We should not overestimate or exaggerate the situation there.
So are you going to contact the journal editor to withdraw the paper, or the first author or corresponding author? No, I'm joking. But what happened is last year, there are a lot of papers were withdrawn from high-profile journals because they used bioinformatics tools and they publish it. A lot of high-profile journals, and because of the bioinformatics tools were developed without validation. Right? So we have to speak out. Sometimes if you see this kind of bad result, totally misleading, we have to speak out.
We can do it together if you want.
Okay, that's my comment.
So because our time, we are jumping for the next lecture, okay? So thank you, Amin, again. Nice presentation. And for the next presentation, I would like to invite Dr. Jamel from University of Lille from France. So, stage there. Jamel, do you want to? Want to sit?
I don't know. I think I just how it works. This
Laser points here.
Yeah.
Forward, back.
Okay. So, hi everyone. Bom dia. I have to apologize because for my English, which is a bit frozen. As you know, in France, we do speak French. So, I'm going to do my best to make my English clear. A story, I stayed in US when I was young. I thought to improve my English. But in New York, everyone was speaking Spanish, so I improved my Spanish rather than to improve my English. So the presentation I want to include today in this nice seminar is going to be on enterocin DD14, which is leaderless bacteriocin, and it's another window, another way to see antimicrobial resistance interventions, etc., etc. So, before starting, first I'd like to thank this gentleman, Nehru, we have a great interaction through students. We have also university agreement between Lille and the University of Viçosa. You know, I like Viçosa. At the point, I gave part of my blood yesterday. So it's okay. So, Neru, muito obrigado. So, just to show you that I come from this lovely country, which is France. And just to make online, here is Lille. So, it's close to the Belgian borders. When we invite people, we don't tell them, come to the north of France. We tell them we are close, we are in the south of Belgium. So this mitigates the, the temperatures. Lille is actually well located geographically because we have 1 hour by train to get in Paris downtown. We are not far from London. It's about 1 hour and 30 minutes to get in London Saint Pancras. And for me, I'm Algerian native, so it takes me 2 hours to go back home. So the University of Lille, historically, it's the University of Lille is the hugest one in France. It's about 80,000 students and 9,000 people working there. And historically, it started to be built by Spanish King Felipe II. Yeah, you didn't know that. I think you should go online and to see these informations. So, what to say more? That I think we have some data. Maybe to say that Pasteur also, Louis Pasteur, he was a dean also in the University of Lille. Okay, the content that I'm going to detail today comes basically from this review that I gave to the students. This review is basically or fundamental, dedicated to this bacteriocin because we have done like 15 papers just to understand this bacteriocin. So I will gladly share some of them with you today. So I have a question for students. Usually the first question, if I show this picture, do you know? Who is it? Angelina Jolie, très bien, good. This one? Georges Clooney, excellent. Let's keep. Wow, that's fantastic. So you have your exam and we can go back home. Now we will show you this. You know him? Things are becoming tough. I will show you. He found this molecule. Okay? He got a Nobel Prize. You know who is this? Okay, I will help you. By this second, do you know him? No. He found this molecule. He was Nobel Prize too. Okay, I will send another one. I'm pretty sure Paul, he knows who is. Because he was from Scotland, he was born in Scotland and he died in UK. He found this molecule and he got a Nobel Prize. So you figure out who are these? I will give you the response. The first one was Gerard Domac, who discovered these antibiotics, which is called Prontozil. The second one was Selman Waxman. He found streptomycin. And obviously, Sir Alexander Fleming, because Alexander Fleming, I should mention that he likes soccer. Okay, so I'm not going to say a lot of things on the outlines regarding the content, because my colleagues, the previous speakers, they have done bright introductions on the topic. Just we can keep in mind that indeed AMR is a big issue, is one of the top global public health in the world. This data, you got them, even you got very fresh data from Amin. So just we keep in mind that AMR is a problem. As we are dealing with, this is for students, and I agree absolutely with Valentina when she said yesterday that bacteria are intelligent. I do believe, because they do have a lot of strategies to cope and to behave very nicely in different environments. So this is just a review article that I took from Nature, just to say to the students, you can go through it and see that bacteria can set up different strategies to face the antibiotics. Without getting in detail, you have the active efflux, downregulation, inactivation of antibiotics, and so and so. So definitely, bacteria are intelligent. So if we come to the biofilm life of style, Valentina mentioned, and the problem of biofilms, indeed, I mean, the biofilms that do not help the antimicrobial treatment. One point, so they can decrease the permeability, they can act against when bacteria get living in such a structure. So they can decrease the permeability, they can lower the expression of target proteins, and they can also have to give a chance to persisters to wake up or to develop. Here you have the definition of persisters. Another thing, that when you have this kind of structures, it does mean that you have movement of IMR genes, and this helps to spread easily the DNA determinants. Another point that could be added is the fact that to have bacteria with such exposure, so they can lose their cell wall and become like mycoplasma, they become like L-form bacterium, so longer they are not resistant to, for example, to beta-lactams. That's the first point that I wanted to share with you, mainly with students on this point. Okay, now let's move to the development of new antibiotics. It's everyone will agree that looking for new antibiotics is just like looking for a needle in how we say that it's really complicated because it's a big effort and you have a 99% of chance to fall. Lastly, I mean, from 2014 to 2021, only 18 antibiotics have been approved. So, if you want really to see the reasons why there is not a big business, and the reason that we are not doing research on the new antibiotics, I suggest to have a look at this review, which explained very nicely these reasons. So now, the development of new antibiotics is becoming a problem, but we have to see two alternatives that could be set up. You have a phage therapy, bacteriocins, you have probiotics, fecal microbial transplants, and you have monoclonal antibodies. I'm going to speak on bacteriocins, but I wish also to add my print that antimicrobial resistance increases, and there is a nice paper which bring the artificial intelligence as maybe one option and one solution to mitigate this problem. So now let's move to bacteriocins. Bacteriocins are antimicrobial peptides or proteins that have been discovered in 1925 by Professor André Grazia. If you see, it's almost 1 century, and it was 3 years before before the discovery of penicillin by Sir Alexander Fleming. So bacteriocins are produced through ribosomes by gram-positive, gram-negative, and archaea, with a size ranging from 9,6 Dalton to even 7,000 Dalton. Usually they are cationic and hydrophobic peptides, active against bacteria which are phylogenetically close to the producing strain. I do mean if the bacteriocin comes from gram-positives, most likely it will be active against gram-positives. If it comes from gram-negatives, so it will be active against gram-negative. But in the lab we have found a strain from gram-positive bacteria, which is called Lactobacillus paracasei, and this strain is able to produce even 5 bacteriocins with activity against E. coli, even E. coli with resistance to colistin. Currently, there is not a universal classification and common scheme which is accepted by everyone. So the classification of bacteriocin is always evolving, taking into account the last advances and progress made in the field. We think we have data that the transport of bacteriocin from the cytoplasm to the outside of the cell is most often performed by the ABC transporters. And few examples, few cases of bacteriocin, they do go through the SEC-dependent pathway. But in the lab, we have found another system that I'm going to highlight on it in the next coming slide. So bacteriocin are not only antimicrobial peptides. They do have activity against viruses. They can be used as potentiating agent to help the activity of aging antibiotics. They can be regulator of microbiota. They do have some anti-cancer activity. They do have, they could be like plant growth promoters, and obviously as food additives. So in this case, we can say that they have a multifunction, they are multifunctional. Here is, I imagine, like a knife from Switzerland. So in this, it's not working anymore. Okay, so in this context, we have published a couple of reviews with Michael Chikindas from Rutgers University, in which we show that bacteriocin indeed are very interesting compounds to be studied in these fields. So now for the student, I will highlight how the bacteriocin is produced and how it moves from the cytoplasm to the environment. Usually a bacteriocin is produced from different, the bacteria mobilizes different genes to produce its bacteriocin. You have usually a structural gene, and the bacteriocin is produced like a pre-peptide. The pre-peptide is not really active or even weakly active inside of the cytoplasm. So when it comes outside of the cell, here you have a possibility to make post-traductional modification, and we get in different types of bacteriocins. So what's to keep in mind that the signal peptide helps to keep the bacteriocin inactive and helps to drive the bacteriocin to the transporters. And hopes you have a cleavage, a cutoff, and the bacteriocin becomes structured and active outside. There are also bacteriocins which are not endowed with, which do not contain these signal peptides. So they are produced basically by this similar machinery. Then you have a peptide which is active inside of the cell, and the bacteria has all the right to expel it outside very fastly because inside it's active. This is the production, the transport of bacteriocin of prepeptide versus leader lyase. So the bacteriocin we have studied deeply in the lab is a leader lyase. Just for your information, here are some signal peptides. It can be classified in different groups. The signal peptides, just a message to take home, is involved in the export of the bacteriocin, in the recognition of bacteriocin by the modification machinery, and it keeps the bacteriocin weakly or even not active inside the cell. And a paper coming from USDA a long time ago. So these colleagues, they put some peptide to increase the amount of bacteriocin produced by some lactic acid bacteria from Enterococci. They were adding either this peptide extracellularly, these peptides, signal peptides, and they could really increase the amount of bacteriocin, the bacteriocin. So now, if we look at the effect of bacteriocin on microbial communities, I suggest definitely to take a look at this paper from our colleague and friend Simon Heilbronner. He's in München University. So in that paper, he explained very well that production of bacteriocin, they can facilitate community invasion. So we can say there is a niche clearance. They can also make preventing community invasion, so they do a colonization resistance, and they can prevent community differentiation, so we speak on spatial segregation. Now, how bacteriocin do act on the target strain? You heard this morning antibiotics. We know basically how they work, antibiotics. So the bacteriocin, when they come from gram-negative, so they will recognize a receptor. So they stick to the receptor. This is a bacteriocin from gram-negative. With the help of a translocator, they come to the periplasm. And then with specific proteins, they can, here you go, here you go. So then inside, with the help of toll or ton proteins, so they can make a pore and get inside of the cytoplasm and make their job inside. So now if we look at the bacteriocin coming from gram-positive, they are active against gram-positive. Basically, they do act at micromolar, nanomolar, even picomolar concentration, which is an incentive versus antibiotics. So if they can form pores, or they can, for example, if you take the case of nisin, which is well-known bacteriocin, nisin can stick to lipid 2, and then it prevents the elongation of peptidoglycan formations. So it just this to pay your attention that bacteriocin, they do have some similarities with antibiotics in terms of mode of action. Here is the review that we have published with Ismail Fliss from Laval Université, Paul Cotter from Ireland, Sylvie Rebuffa from the Museum Histoire Naturelle in Paris, in which we give details on the mode of actions of different bacteriocins in both models, in gram-positives and gram-negatives. And you can take a look at this nice review. Now, I will explain, because we want to come with solutions to interventions. I mean to say that we have a nice molecule and I have a nice project, but in my brain, we need to understand how the whole system is working. That's why I'm going to give you some details, how gene regulation is done in Enterococcus before coming to enterocin, enterocin DD14 before coming to its application. So the story of enterocin DD14 is almost 10 years old. We started within a PhD project to take samples of meconium in Roubaix Hospital. It's close to Lille. So we started to take samples randomly and to see if there are lactic acid bacteria here, and if there are lactic acid bacteria in the meconium, to isolate those which produce bacteriocins. Meconium is the first dropping from newborns. It can be greenish, dark, but sticky. So at that moment, it was something just to gamble if we have lactic acid bacteria there. So in fact, we have isolated one strain which is called several strains, but one of them is called Enterococcus faecalis 14. This has activity against gram-positives, mainly Clostridium perfringens, and I do have data, very nice data on this, but unfortunately today I'm not going to speak on them, against Listeria, against other Enterococcus, even against Enterococcus V583, which is a well-known strain. This work was done with Ahmed. He was from Iraq, PhD student at that moment, and now he is back home where he is like professor in Tikar University in Iraq. So then we hired Delphine Kali. She was a very good postdoc. She started to study this bacteriocin and to identify, sequence, etc. And we come to the conclusion that this bacteriocin is a leaderless, is composed of two peptides, which called the DD14A, B, and one of them is 44 amino acid, the other 43 amino acids. And this one of these or her papers, at least this one was published in International Journal of Antimicrobial Agent. So now for the student, what I showed now on leaderless bacteriocin. So leaderless bacteriocin, they do not have signal peptide. They are active upon their translation in the cytoplasm. Usually they do have a formyl methionine instead of a methionine. They do not go post-translational modification as some RIPs do. And they do have a motif which is called the saposin-like motif. The question which remains open on leaderless bacteriocin, what is the system, which protein is involved in their transport inside the cytoplasm to reach the transporters? How the immunity is ensured, I mean, which is the system, the proteins or whatever which is involved in the immunity of the strain producing this leaderless, and what is the role of formyl methionine in the global antibacterial activity. So what we have done then with Yanat, we sequenced the genome of this Enterococcus faecalis 14. We did some bioinformatic analysis using software like Beagle 3, then Beagle 4, and we come to locate these genes. Here you have a cluster with two operants. The first one is composed of gene A, gene B, which are controlled by promoter, which was called P1. And here you have a second promoter, which controls the remaining genes. So here it fits very nicely with data previously obtained by Delphine. So Yanat is still in the lab like an engineer. So the question, at the fundamental academic scale, we wanted to know if this bacteriocin was the only compound responsible for the antibacterial activity. We also wanted to know if this strain, Enterococcus faecalis 14, when it is not longer able to produce this bacteriocin, what happens? And then we tried to understand how this bacteriocin is transported from the cytoplasm to outside of the cell. So if you look at this third question, if you look at the genetic organization of this cluster, here you have a block of ABC transporters. So naively, we can say, oh, you have a typical accessory protein, you have a flax R&D system, ATPase, you have a permease, etc. So if you look at this, you can say it's coming through this ABC system. No, it's not completely through. I will tell you why. So the first thing that we have done, we were very lucky because Enterococcus faecalis is easier to genetically modify. We have done build some isogenic strain using a technology taken from this paper. We could make inactivate or remove any gene, even a gene located in the operon, without any polar effect. It comes with two genetic events. I mean, this is the system that we have used to remove genes and understand the global function of this operon. So the first mutation that we have done it with Rabia. He was like assistant professor for 4 years. With the precious help of Anka, she is here today. And what we have done, we started. This is the full operant. We removed the AB gene. It does mean the genes that codes for the bacteriocin. And then we do not obtain any activity, even though the qPCR analysis showed expression of these genes. And we could also detect the bacteriocin inside of the cytoplasm. So that's the first achievement, which was very important for us. So then, with the help with Anka and Rabia, we did some microarray analysis in the delta BAC mutant. It does mean the mutant which not produce longer the bacteriocin. And we found like 69 genes which were upregulated. And as you can see here, these genes were involved in virulence mechanisms, phage-related genes, and so on. So genes involved a lot of biological function. It does mean that this strain, when it loses its capacity to produce its weapon, so it gets like scared and start to modify its metabolism to produce, to make other strategies. So a message to take home, the delta BAC do not produce, and interestingly, the delta BAC is sensitive to its own bacteriocin when you provide it extracellularly. And we have published a paper in Frontiers Biotechnology. Second mutation, we did it with still Rabia and Adrian. He was a Spanish postdoc from Madrid, and just his PhD supervisor was my postdoc supervisor. So you understand the connection. And we have done the first mutation. As you can see, the first that we removed part of the ABC transporters. I mean, the permease function was deleted. Then when we measured the activity of the bacteriocin, 25% of activity versus the wild type. So when we remove all the block, we still do have 25%. It does mean that the bacteriocin, at least this one, the leaderless, is not necessarily using the ABC transporter. There is another system. So this was okay. We were in this configuration, I mean in the absence of the ABC transporter. We do believe there is a channel composed of DDE, DDF proteins that helps to bring the bacteriocin from the cytoplasm to the outside. So, to prove that, what we have done, we have done a third mutant in which we removed the E, the F genes, which code the E for protein DDE, DDF for the protein, F for the protein DDF, which are membrane proteins. They are cross-membrane proteins. The only gene which is not in the membrane, which is coding for something which is inside, is the protein C. So when we got this mutant, just to tell you that no activity was, when you delete these genes, you do not have any activity. Some bioinformatic analysis told us that this protein holds a PhD domain, which is plexin homology domain. It's about 100 to 120 amino acids. Most often they are found in eukaryotic cells, where they do play a role in signaling, cytoskeletal organization, membrane trafficking, and phospholipid processing. The question if this PhD domain comes from eukaryotic or from bacteria, it's still, for me, a debate. So we started to do, we focused then on this channel. This is a mutant DDE, DDF. When you remove either one or the other one or both of them, no activity outside of the cell, no bacteriocin outside of the cell. So the bacteriocin gets stacked in the cytoplasm. We could detect it by mass spectrometry, and we registered in both mutants 1 log death after 24 hours. So here you go. So this is another work that we have done just to make models of these proteins. DDF is constituted of 6 subunits. This is when you look at it from the outside. Here it's in the transmembrane, and here inside. As you can see, something is missing. Here is the DDE with 2 subunits. So when you take both of them, you make 90-degree rotation. It clips and forms these channels. So the channel is likely in this way. Okay. Come on. Okay. So the hypothesis on the transport of this bacteriocin. So when Enterococcus, as you can see, is here, is very happy because the bacteriocin, the whole system is present. So the bacteriocin most likely go first through this channel, DDE, DDF, and then it switch somehow to the ABC transporters. But we need to work also on it. So if you want, as we say in French, la science n'est pas une vérité, mais une vérité en construction. Science is not a truth, but a truth under construction. So we have to work on it. We published a couple of papers, but a lot of job is remaining to be done. Oh, I will bring you to Algeria if you wish. I'm sorry. So if you have plans for vacation, or this is North Africa. So I was born in this village a long time ago, not very far from this street. So also in Algeria we do have Sahara desert, very huge and very beautiful. We have also La Plage, the village, mountains. We can offer you these things if you come to visit us. So let's get back to the last part of this presentation. It's a paper that we have published recently in Scientific Reports, just to understand how this system is working. So we decided to make another mutant. This mutant is called PNPase, deleted in the gene coding for PNPase. What is PNPase? PNPase is basically an exo 3', 5' ribonuclease, which was discovered by Spanish Nobel Prize, Severo Ochoa, who showed that PNPase was involved in RNA synthesis. So the idea, the PNPS has this activity showing mRNA from 3' to 5'. PNPS has also a polymerase activity. And PNPS is present almost in all lineages. So if you take a look at this paper, so PNPS, not merely an RNase, but also a central post-transcription regulator. What's the idea to do? To link the bacteriocin and PNPS, just to know if we can increase the amount of bacteriocin and in order to produce more and more. What we have done, this is the gene coding for the bacteriocin. We have removed this gene and got the delta PNPR mutant. Here you have it. And this is the wild type, and this is the mutant which is complemented. What I can say, this is the colony. This is the colony becomes smaller. When you put this gene in trans, the strain, the mutant, recovers its the normal situation, and you have colonies similar to those of the wild type. And here you have some microscopy electronic, sorry, and showing the difference and the damage made by Enterococcus faecalis and the wild type on Listeria. Another point that need to be mentioned, we did also some microarrays and published a paper in Gene with ANCA and Rabia and showed that in the absence of PNPS, in the global genome, we had like 200 genes that were affected by the absence of this enzyme, major enzyme. These genes that were affected could be linked to cell envelope, pathogenesis, whatever. Concretely, we have also shown that there was some activity, proteolytic activity that were increased, adhesion to Caco cell. We also found that expression of bacteriocins, or at least gene coding for bacteriocins, was increased. That was the top. We could confirm that production of bacteriocin in the mutant was, in the delta PNPR mutant, was at least 2.5 times higher than the wild type. So, to almost finish, here you have the whole genome. Here you have the P1 promoter governing the AB gene. Here you have the P2 promoter governing the remaining part. Here you have transcriptional terminator, and here you have another one. So with Paloma, she was my postdoc supervisor in Spain. She just retired. I was last week for this celebration in Madrid. If I'm here today, speaking on RNA, speaking on gene regulation, because of this scientist. Thank you. So, what we have done with Paloma, we predicted these terminators. This one, as you can see, is weak. This one is strong. And this one, it is supposed to protect all these genes. And this one is supposed to protect only A and B. So what we have done then is to establish that we have this transcript doing some cDNA analysis. So we extracted RNA from the strain, we did some PCR, cDNA, etc. We could indeed demonstrate that we have a transcript which corresponds to the A and B, and we have also a transcript coming from the promoter until the end, until the transcriptional terminator. Then, that's the prediction. This is the experimental data which fit. And what we have done is to grow the wild type, the mutant deleted in the PNP gene, and to look at the stability of mRNA coding for the A and B for the bacteriocin. and the stability of mRNA coding for this gene and for this gene. I think it was enough, and it was a good proof to come to this paper. So how to study the mRNA stability? You grow your strain until the mid-lag. You add rifampicin and nalidixic acid, and then you take samples at regular intervals of time, 5, 10, 15, 30, and 60 minutes. And you do the same thing with same sample, and then you see the expression of gene coding of these genes. You see, for example, in the wild type, we could detect the expression of this gene even 60 minutes after blocking the transcription. And we got more mRNA after 60 minutes, which proves that the T1 T1 terminator was very strong. If you look at this, in this case, this transcriptional terminator was very weak. We couldn't detect this transcript in the wild type because PNPase is showing this mRNA. So we have now the evidence that PNPase is playing a role, and this terminator are, as predicted, strong and weak. So now let's move to the application of this bacteriocin. We heard this morning very nice presentations, on one side on Staphylococcus and the other on E. coli. What we have done is just to test this bacteriocin against MRSA strain isolated in France. So the question, do bugs need drugs? I hope not all of them. So if you look at Staphylococcus aureus methicillin resistance, it comes like a second priority from the World Health Organization. If you look at the ESBL, they are in the top priority. Nevertheless, we need to find some alternatives. What we have done with IANAT, we purified the bacteriocin. We did some combination between bacteriocin and methicillin. As you can see here, you have the mixed value. So the bacteriocin is almost not active against the Staphylococcus aureus that we have used. The methicillin, you have huge mixed value. And when you combine both of them, you decrease this mixed value. And here you have a killing curves, a typical killing curves, which are strengthened with confocal microscopy analysis. So this is a point. It's really interesting because the bacteriocin at the beginning has no activity against this strain that we have used and against USA 300 strain and et cetera, et cetera. So what to improve the activity of this bacteriocin? We were able to produce some nanoparticles, alginate nanoparticles. Why alginate? Because it's eco-friendly, and alginate has not intrinsic antimicrobial peptides. So when you load bacteriocin on alginate, you have some activity. When you decrease, you improve the mix value. So when you put on these particles, you put your bacteriocin plus methicillin, so we improve the activity, obviously. Here you have. Then we tested the activity against biofilms. This is done on glass slides, the biofilms, and we tried to see. Here you go. You have the activity against biofilms. So the combination is not. Is promising, but it's a proof of concept, and we need to keep in mind that this strategy could be an issue. We looked also at the expression of gene coding for virulence factors. That's why I appreciated the question from Paul coming this morning. So when you have the bacteriocin in contact with Staphylococcus, so you down-regulate some virulence factors like staphylococcal nuclease or
PVL, which is ponton-valentine leukocidine. And this is perfectly in the strategy to find new drugs for bad bags. And these new drugs have to be anti-virulence strategies. Maybe one is to look at these things. Then we did some in vivo assay. What we have done on this work is to design small groups because it was a bit complicated to do at that time differently. 5 mice by group. This is the control. Control treated, infected with our strain. Here, this is the control. This is the mice infected with the same strain, but for which we have added erythromycin. The strain I mentioned is resistant to erythromycin, but when you put erythromycin and bacteriocin in vitro, the strain recovers, becomes sensitive. Here, the group number 4 received the strain and the bacteriocin. You know, the bacteriocin is not active, and the last group has received erythromycin plus bacteriocin. So the experiment was monitored for 4 days with all the ethical aspect, with the help of DVM, and the molecules were injected through the peritoine, and then we used this holozenic naval, this line of mice. Then we did some histological analysis. I'm not going to detail, but if you look at the crypts are here very well in the colon. They were very nice also in the colon, and also, I'm sorry, yes, in the group which has not received nothing, the crypts are okay. The groups that have received the bacteriocin and the antibiotics, they are okay. And the groups that have received only the bacteriocin, they are okay. You have also more details, but these details, you can find them in the paper. We did some body weight evolution with statistical analysis, and everything enabled us to say that the combination do not alter the microbiota and helps to recover very easily. Here you have. So the achievement on this bacteriocin. So the ABC transporters are not the unique transporters of leaderless bacteriocin. Leaderless bacteriocin are active in the cytoplasm. These bacteriocin are endowed with antibacterial activity, but also with antiviral ones. I will show one slide. And they do have some immunomodulatory proteins. What is left and the next step? Next step, I want to understand how the bacteriocin is transported from the cytoplasm to outside. I do suspect that protein C, which is the only intracellular proteins, is playing a role. So far, what we have done is to make a mutant deleted in protein C, and this mutant do not produce the bacteriocin. Even you have expression of the other genes. It is cloned. The gene is cloned, and the protein is waiting for me in the urea just to understand and do something else on it. Here, almost done. We have shown that this anti-inflammatory effect of enterocin DD14, and the paper has been published with Benoit. We have also an activity on antiviral activity, mainly against enveloped viruses. And we tested in the Hospital of Lille Medical School against herpes simplex viruses, even SARS-CoV-2. SARS-CoV-2 was the first strain that was circulating in the world. So we have some, because I want to, this is the interaction that we have predicted between the bacteriocin and the receptor of SARS-CoV-2, which fit very nicely. You can see the models, but I don't know how the system is not, why it's not working. You know why? ¿Puedes? Por favor. Here, if you can. Yeah. Yeah, that's okay. This is the prediction between the receptors and the bacteriocin. We have done some antiviral assessment, and we submit a few days ago a paper for publication. So the chemical synthesis of bacteriocins, it's really a big a breakthrough, because you know, when you produce bacteriocin, you always have a very low amount. So we were very happy because we could produce this enterocin DD14, these two peptides, which function very nicely. And an upcoming paper is scheduled with colleagues from l'Université of Laval, Eric Biron. So I'm almost done. This is all the colleagues, friends that have been somehow associated to the DD14 story. There are a lot of people, so I'm here. The one I want also to thank a lot is Karima. She is my wife. She is MD. We have two papers together, so we have two daughters together. And that's it. So there are a lot of people. Anne Vacher from Roubaix Hospital. We have Rosen, she is here. Benoit, Onka. Onka, I didn't miss your picture now? You are somewhere here? Okay, a lot of people. So if you want to have your picture on this nice board, we have to collaborate. So just to finish, doing this work was possible through grants. I mean, we got the money from the l'ANR, Le Savre, l'Université de Lille, so they paid the bill. And to finish, we are doing our best to understand microorganisms, but Pasteur was saying, ce sont les microbes qui auront toujours le dernier mot. It does mean that microbes will have the last word. For this, we keep it here today. Thank you.
So we have questions here.
Thank you so much for the presentation. I'm going back to one of the first study you show about meconium. Sorry, the infant.
Yes, meconium,
okay.
Yes.
So my first question is, how did you come up with testing that stool? And then is there a follow-up study where maybe you can follow the infant for the first 6 months and then compare breastfed and not breastfed? Then maybe you can see a diff. Because I think what you are trying to demonstrate, if it's coming through the mother, maybe in the meconium or not. So thank you.
So thank you, Valentina. There are actually three questions. I mean, because 10 years ago, when we started this project, we thought that meconium was something that was germ-free, sterile. But if you look at different papers then published on meconium, you have a huge community. So the idea just was to see if we can recover lactic acid bacteria from that matrix. And then we didn't go far in terms of understanding the evolution or something like that. Just to take like first step, isolate, and see if we have strain producing bacteriocin. But indeed, I agree with you. There are a lot of people working now on meconium, and there are bright labs in US mainly. They do the ecology of meconium, and there are a lot of things to be done. But and also, the reason also this was a little bit personal, because my wife was working in the same hospital from where we took the samples. Said, okay, let's do something together. We did this. But what I'm tempted to suggest to the future Mama, don't trash your meconium because you can have strain with very, very important value. So just hold it, and maybe you can do some PhD project, and that's it. Merci.
More question?
Good morning.
Hi.
Thanks for the presentation. It was really good. So, as you said, we have nowadays a lot of problems with the resistance, antimicrobial resistance. And at the same time, we have the development of the alternatives like the bacteriocins that present a probiotic potential. Yeah. So, we can already apply this alternative in the practice, or we still need more researches and studies. Also, would the cost be too high for the industries?
So thank you very much for this question also. I think there are two questions. One is the alternatives, the application of bacteriocin. As I mentioned in the beginning, we try to provide proof of concept that it can work. Okay, it can work. But get them in the market, and there is a regulation which is very demanding, very demanding. So the problem is the regulation, and you need also to show the development of resistance between bacteriocins and the antibiotics. These days I'm thinking to a project, very focused project on this topic. The second question, which indeed is, when you produce a bacteriocin from a single strain, it's really cost effective because the strain is producing very low amounts, even though it has to mobilize a lot of genes, a lot of determinant genes to finally produce a little amount. Why? Because when the bacteriocin is produced, as I mentioned, it can be active at the picomole quantity. The strain has not to produce big quantities. But if it's active with such concentration, you can also take it like an advantage versus the antibiotics. Okay? So to produce more, you have different way to do it. Depends if we go to the food science or to medical science. If you want to produce more, you have to do some production in host systems in which you control the production, heterologous production. And the chemical production is now becoming serious because when we started like 10 years ago or something, it was very hard to produce, for example, a pediocin like bacteriocin because they have two disulfide bridges, but now they are very easily produced. So the production through chemical synthesis is also here to improve this amount. Yeah. Thank you.
First of all, thank you for the presentation. And my question is about if there are any records about resistance against the bacteriocins, or if there are any resources about this, if it is possible, because if it's an alternative against the AMR, if we can get microbials that are resistant against the bacteriocins.
You know, I have to be back to what mentioned by Valentina. The microorganisms are very intelligent. I mean, even though you give a bacteriocin, you will end by generating the resistance. But the resistance, it's also dependent on the concentration of the molecule that you have been using. And in case of bacteriocin, the scale of development of resistance is less, in my opinion, important than those seen for the chemical molecules. But experimentally, you can prove it. And the idea, you're right, is to combine maybe to not put, this will join the former question, to not use bacteriocin, but to use bacteriocin and antibiotics, so you can decrease the amount of antibiotics, you save their life, and you keep their effectiveness. Maybe the combination, in my opinion, is something that need to be more examined. And for the application, at least at medical things, this is an option for me, which is very serious. Paul, you have a question?
Well, I was going to ask actually whether you're tested the synergy between your bacteriocin and antibiotics, because obviously you did the AN experiments. I couldn't see it, but it sounds as though you haven't. But have you, or have you done those experiments?
For what?
To where you have the antibiotic and the bacteriocin together.
Yeah.
Have you tested that? Is there synergy?
Yeah.
There is?
Yeah. We did it with different bacteriocins. We did it in this case. We did also with colistin, because we have tested in a ESBL strain, MCR strain, and when you combine bacteriocins and these antibiotics, you decrease the mix value. But for the moment, we need also to go to the animal models.
Yeah, that's what I was going to ask, because obviously
it
doesn't always pan out with those, but yeah.
Yeah, it's in the pipeline. I mean, the project is in the pipeline. We applied also for staff project justly to have something similar. Anyhow, thank you. Yeah. You are not fair with me. It's a lot of questions in my state.
Jamal, thank you very much for your very comprehensive work. I really appreciate your 10 years of work for Anderson. Yeah. So I have a question about your alginate nanoparticles.
Yeah.
When you make the alginate nanoparticles, how did you make it? Did you encapsulate or you conjugate the
No, we have an Institute of Nanotechnology close by. We have a system, I mean, to prepare this by ourselves. Unfortunately, I didn't prepare a slide on it, but I can tell you very easily how to do them.
So it looks like you did encapsulation to enhance the dilbel.
Not encapsulation. Yeah, once we have the nanoparticles, we load the bacteriocin on it. It's not covered.
Okay.
It's like interaction that's between the bacteriocin positively charged, and we have the alginate, and then you can keep the surface of the nanoparticle with some molecules of bacteriocin, and this helps to improve the activity. You can load a certain amount.
So I believe you heard about the microcin J25. That's
of
also
course.
Another, yes, of course. So we recently published a paper. We conjugated microcin J25 to cytosine nanoparticles.
Oh yeah.
Then it enhanced the antimicrobial activity about tenfold.
Yeah.
I may talk with about that later.
My pleasure.
Sure.
Is that bon?
Any more questions? No? Okay.
Muito obrigado, and thank you so much. Thank you for helping me, because I got very good assistance with Emily, with Nero, because yesterday, as I mentioned, I gave some of my blood to the university. Thank you.
Thank you, Jamal. So I see you, everyone, tomorrow, okay? Thank you.